Hello everyone. This video is about Chapter 9, Lesson 2, the process of cellular respiration, from page 254 to 257 of your textbook. Our objectives: at the end of this lesson, we should be able to describe what happens during glycolysis and describe what happens during the Krebs cycle. So as you all know, we get our energy from the food that we eat. How does a living cell extract the energy stored in food without setting a fire or blowing things up? Of course, this is through the process cellular respiration. And cellular respiration has three stages. But I'm only going to go in depth into the first two stages for now and have another video for the third stage. So we'll start now with the first process, and that is glycolysis. Glycolysis is the first stage of cellular respiration. During glycolysis, glucose is broken down into two molecules of the three-carbon molecule pyruvic acid, and pyruvic acid is a reactant in the Krebs cycle. ATP and NADH are produced as part of the process. So as you can see in the figure, you have there the production of ATP and NADH in the process of glycolysis. So during glycolysis, one molecule of glucose, a 6-carbon compound, is transformed into two molecules of pyruvic acid and three carbon compounds. So what is ATP for? ATP is both used and produced in this process of glycolysis. The cell deposits two ATP molecules into its account to get glycolysis going. So you've seen there that the ATP is being used up to use or to produce ADP. Glycolysis then produces four ATP molecules, giving the cell a net gain of two ATP molecules for each molecule of glucose that enters glycolysis. So again, at first, it deposits two ATP, so it uses up these two ATP, then produce 4 ATP, and so the net gain will be 2 ATPs at the end of this stage. During glycolysis, the electron carrier NOD or NAD or nicotinamide adenine dinucleotide accepts a pair of high energy electrons and becomes NODH. NODH carries the high energy electrons to the electron transport chain, where they can be used to produce more ATP. Two NODH molecules are produced for every molecule of glucose that enters glycolysis. So at the end of this process, what are our products? We have two net gain of ATP, and then we also have two NODH or NADH at the end of this glycolysis process. So what are the advantages of glycolysis? Glycolysis produces ATP very fast, which is an advantage when the energy demands of the cell suddenly increase. Glycolysis does not require oxygen, so it can quickly supply energy to cells when oxygen is unavailable. So glycolysis is an anaerobic type of respiration, and we all know that when we say anaerobic, that means it does not require oxygen in this process. The second stage of cellular respiration is Krebs cycle. So what happens during the Krebs cycle? During the Krebs cycle, the second stage of cellular respiration, pyruvic acid produced in glycolysis is broken down into carbon dioxide in a series of energy extracting reactions. The Krebs cycle is also known as the citric acid cycle because citric acid is the first compound formed in this series of reactions. So this pyruvic acid from the glycolysis enters the matrix. What is a matrix? Matrix is the innermost compartment of the mitochondrion. So once pyruvic acid is in the mitochondrial matrix, NOD+ accepts two high energy electrons to form NADH. One molecule of carbon dioxide is produced. The remaining two carbon atoms react to form acetyl-CoA. So from a three-carbon compound of pyruvic acid, one carbon is released in the form of carbon dioxide, and this two remaining carbon dioxide becomes acetyl-CoA or reacts to form acetyl-CoA. This acetyl-CoA combines with a four-carbon molecule to produce citric acid. So again, from a two-carbon compound, it reacts with or it combines with four-carbon molecule to produce citric acid. So that becomes now six-carbon compound. So citric acid is a six-carbon compound. So citric acid is broken down into a five-carbon compound and then a four-carbon compound. Two molecules of carbon dioxide are released. The four-carbon compound can then start the cycle again by combining with acetyl-CoA. So as you can see from the figure, from a six-carbon compound, it releases one carbon dioxide, reducing the six-carbon compound into five. Then again releases one carbon dioxide, reducing this five-carbon compound into four-carbon compound. And again, this compound will be used to start the process again. So a Krebs cycle is a cycle because the four-carbon compound produced in the last step is the same molecule that accepts the acetyl-CoA in the first step. Energy released by the breaking and rearranging of carbon bonds is captured in the forms of ATP, NADH, and FADH2. So as you can see in the figure, the orange icon is the conversion of ADP into ATP, and we also have there the FAD into FADH2, and lastly the NOD, which is converted into NODH2. So for each turn of the cycle, one ADP molecule is converted into ATP. This ATP can directly power the cell's activity. The electron carriers NOD plus and FAD each accept pairs of high energy electrons to form NODH and FADH2. NODH and FADH2 are used in the electron transport chain to generate more ATP. So that's it for the first two stages of the cellular respiration. Let us now summarize the first two stages of cellular respiration. For glycolysis, the word glycolysis literally means sugar breaking. The end result is two molecules of a three-carbon molecule called pyruvic acid. Two ATP molecules are used at the start of glycolysis to get the process started. High energy electrons are passed to the electron carrier NOD, forming two molecules of NODH2. Four ATP are synthesized during glucose or glycolysis for a net gain of 2 ATP. In the Krebs cycle, the second stage of cellular respiration is the Krebs cycle, which operates only when oxygen is available. The Krebs cycle is a series of energy-extracting reactions. Pyruvic acid, which is produced by glycolysis, enters mitochondria. In the innermost compartment of a mitochondrion, or the matrix, pyruvic acid molecules are broken down into carbon dioxide and acetyl-CoA molecules. This acetyl-CoA combines with a four-carbon compound, producing a six-carbon molecule, citric acid. Energy is then released by the breaking and rearranging of carbon bonds, which is captured in ATP, NODH, and FADH2. The Krebs cycle produces four types of products: high-energy electron carriers, the NODH and FADH2, carbon dioxide, two ATP molecules per glucose of molecule, and the four-carbon molecule needed to start the cycle again. So let's now have some lesson check questions. Number one question: what are the products of glycolysis? Can you please think of your answer? The answer is two molecules of pyruvic acid, two molecules of NADH, and a net gain of two ATP molecules. Number two question: what happens to pyruvic acid in the Krebs cycle? The answer is pyruvic acid is broken down into carbon dioxide through a series of energy extracting reactions, collectively called the Krebs cycle. Number three question: what are the products of the Krebs cycle? For answer, the products are ATP, NADH, PODH2, and carbon dioxide. Now for our last question: what happens to each of products of the Krebs cycle? The answer is carbon dioxide is expelled in exhalation. The ATP molecules are used to power cellular activities. The NADH and PODH2 molecules are used in the electron transport chain to generate ATP. So that's it. Thank you for watching. May you have a nice day. Thank you.